So we're going to start with Victor Meru. Victor is a PhD student in John Marshall's lab at the University of California, Berkeley. He specializes in infectious disease modeling. Prior to starting his PhD, he was a data scientist at Ithakar Health Institute supporting the National Malaria Control Program. And he's going to be sharing really cool modeling work about gene drives. Take it away.
Hello. All right, thank you. Thank you, Josh, for the introduction. Yeah, so yeah, as introduced, I'm Victor. I'm a doctoral student at UC Berkeley, and I will present on the impact of gene drive mosquito release on the community immunity and malaria case resurgence. And it's my first time to use this pointer, so please, when it just... Is it on? All right, this one, okay. Okay. So yeah, I'll take you through some background information before we proceed with the gene drive work. So in the recent years, we have been some substantial progress in malaria control. And however, key challenge remains if intervention that's being deployed do not confer immunity. So some studies show that, yes, ITNs, IRS, and other kind of intervention, when be deployed effectively, can reduce malaria. But those kind of intervention, they do not infer immunity, or they do not confer immunity to the human. So most of the susceptible population is being left without immunity. So when it comes that particular intervention is being stopped or not in progress, the tendency is like the malaria probably might resurge, and when it resurge, it has a bigger effect. I think this is the point. As you see from this study, on the highlight gray, if you can see properly, the gray, this is a part of which intervention was done, and soon after they stopped the intervention, you see that the cases, there's a sudden resurge of the infection. And another study also yeah. Sorry for the technology. Yeah. So similar to this other study, both of these works were modeling work, so they tend to show that the intervention, when it works properly, it leaves like community is more vulnerable to get infections once those kind of intervention have been left. And this is due that those kind of intervention, they do not contribute to immunity. So yes, we have now gene drive. I'll just have a basic introduction to gene drive and what it does. So gene drive, the technology that is a genetic modification technology that is being introduced to mosquito, and it can also be introduced to other organisms, but for our focus here is mosquito population. So the gene drive is a modification of parts of the genes on the mosquito that can tend to either reduce the population of the mosquito or replace parts of the gene that can either block those particular mosquitoes from transmitting certain diseases such as malaria. So yes, so on this work, simulation was done with a focus on replacement type mechanism of gene drive, and the focus was Anopheles gambi. And there are some, so this is what the mechanism is being done for instance, for this case, here's the replacement. So what you can do is, this is illustrative diagram to show what is being done. And yeah. So yes, we have the gene drive, and it is not yet released, but it's a technology that promise that yes, maybe it can handle on top of ITN and RS the decrease or eliminating of malaria. But there are still some, since it is not yet released, and there are still some questions to be asked, like, so due to the nature of it, the key concern here that it is a non-immunizing kind of intervention like the ITN and IRS. What would be the risk of resurgence in case? So from these kind of concerns, there we came up with the questions that, like, how does the gene drive mosquito release impact the community immunity and overall incidence in malaria? And the other question is, like, what is the risk of malaria cases resurgence after the gene drive intervention stopped, and how can it be medicated? So this is a work in progress, and I will present some of the results, particularly for question 1, and others can be, since it's a work in progress, maybe I can get comments or any feedback from you. So this is the simulation workflow. These diagrams have been published elsewhere on that citation there. And so this is the flow of gene drive of this work. And so gene drive parameters, these are the gene drive but were being used, parameters that were being used in our model. And these parameters also are being published in another study. So I applied the parameters since they were well calibrated and they are well validated. So I decided to use the parameters. And yeah, so also during the simulation work, we had 2 sites, Bobo de Alaso, Burkina Faso, and Mwanza in Tanzania selected. The reason behind this is they have different transmission setting. So for instance, in figure A, Bobo de Alaso, it shows like its rain profile is unimodal, meaning that it has only single peak of rainfall, while the other one in Mwanza, it has bimodal. And the rest are those, the parameters which were collected from MAP, Malaria Atlas Project. And so the simulation modeling model that was used is MG Drive. So in our lab, we have series of these models called MG Drive. This is a model that is specialized in modeling the release of gene drive in the environment. And yeah, so the MG Drive 3, which is the latest model, is a model that it has both the entoside, whereby the gene drive, so here we see like it has the inheritance part of the mosquito life history and mosquito, and also it incorporates the seasonality. But not only that, it also incorporates the epicide, which it comes from the malaria model from Imperial College London. And so this model handles both the ento and the epi. So that's why I selected it, because it can handle both the dynamics of gene drive release and what will be the effect on the community in terms of the infection and yeah. So from the parameters, then we're all fitted on the model. We had two scenarios in this study. One study was to see what will be the effects of a gene drive that has a short window of protection. When I'm talking about the window of protection, I talk about the coverage on which the gene drive will be effective. So I will walk you through this diagram. So this is our result, which is comprised of the mosquito part and the human part. So the above diagram, this tells us about how well the gene drive was released and what is the effect, what happened. So on this highlighted this rectangle here, this is our window of protection, whereby, as you see, from the mosquito part here, as the homing allele cross with other area, this is like the blue is like the wild mosquitoes, and the red is like now that gene drive, that kind of mosquito that were released. So when they cross means like the wild type goes to 0, and those which are modified goes up. So we expect that when the homing, which are the one we want to transmit, we want to spread them to the environment. When they go up is when that we see the cases, malaria cases, going down. But what we expected, so now, as you see the malaria cases goes down, we also see the immunity also going down, meaning that people are not being infected, people are not being beaten with infectious mosquitoes. So as they tend for a long time, they also, so it's like there is advantage of losing cases, reducing the cases, but also there's a disadvantage of people reducing the immunity level to being dropping down. So we had the scenario one that we see, okay, maybe this is for a short period of window of protection. So this was the result for the window of protection, the short window of protection. But however, we said, okay, maybe let's try to see what will be the effect if we run a simulation for a long window of protection, and to see maybe doing gene drive for a few more years, maybe after withdrawing that gene drive, or what will we see in case of. So these were the results also. So you could see like this went very far, but as time goes, that effect of the window of protection tends to drop, and also the cases rise. They rise very fast. So at this point, these are the results that I have, and so yes. What are the takeaways of this dessert that I have right now? So you see, like, on the, so even also gene drive, like the other studies we saw, like ITN and IRS, still, if you don't continue for a certain period of time, you just sudden stop, there's a risk of malaria resurgence at some point in time. So, and why resurgence happens is due to the effect of the waning of immunity, as people in the community not, if there is no any other kind of intervention that trying to retain the immunity to the community, such as vaccine and other kind of intervention that may retain the immunity to the community. So even if you use this kind of intervention, which they promise to show like they can eliminate or stop the transmission, there is a risk of resurgence. So I will end with like, we need to use this kind of intervention with caution. Like, we need to have a mechanism to retain the community immunity and not rely only on this kind of interventions. Like, they might work for some period of time, but at some point, the work done which was deployed might be not on effect when the resurgence comes. So that's all that I had for this presentation. As I told you before, it's a work in progress. So still much is to be done, but this is what these are some of the results I had for this presentation. So I welcome question, feedback. Yeah.
Thank you, Victor. We have time for one or two quick questions.
Yeah, thank you so much, Victor, for this very great project. I think it's a promising project. I was struggling to follow if there's some clinical work, say clinical trials, which have been done on it, and it is informing your work, or your work is trying to inform that. I was just keen to hear that from you. The second inquiry is what the potential environmental effects, but also like the way we had it for, the community may reject some of these technologies because of fear, because of modifications. Did you factor that in mind?
All right, thank you. Thank you for a good question. So I'll start with the clinical part. Now, as you know, that the gene drive technology is still a new technology. It has been for a while, but it's still in most cases in labs. And now it's like there's a push now to go to semi-field. Then after semi-field, maybe a release in future. So that's why we are doing some modeling works, because we don't have yet the data. So this kind of work here is like to set a ground in order to, is one thing to have a clue and have like something to start with, and it's another one if you don't have at all. So to my knowledge, that gene drive has not yet been released, and the clinical data yet is not available. So that's why we are doing this kind of studies. But it's a good question, because we also need those kind of data to improve our model. So yeah, so we hopefully the semi-field will start, and out of the semi-field, maybe we can get something. So coming on some The other part of people are skeptical about the technology. Yes, there is a fear of the unknown still. People tend to fear this kind of technology, and there is still a debate on the biosafety people and the researcher. And yes, still people need to, the community needs the disease to be eliminated, the mosquito disease and other kind of disease that maybe gene drive could be a technology to reduce or eliminate. But still, yes, there's that. But yeah, so modeling now, for now, modeling is part of the solution to also to be like so kind of studies like this or other kind of studies on modeling can be used as input for the policy and regulation in terms of, okay, if they want to acquire that kind of technology, what will be the regulation? And if they have fear in case of what will happen in future, at least they have some grounds to think, okay, maybe this scenario can happen. So maybe I'll do, yeah.
Thank you, Victor. I think just for time, we're going to move on. Okay. Those results were very sobering about the loss of immunity. So thank you for that.
Thank you.